Hello, welcome to the first of three videos on mitosis and meiosis. And what we're really going to be talking about here is the idea of cells dividing and developing. So this picture at the top shows you one cell becoming two cells, and that is a process that happens inside of practically every living thing on a second by second basis. Since I've started this video, it's happened in your body 1,000 times. It is happening over and over again in all living things on Earth, and we need to study that process. So the sunshine state standard that goes with this, there's actually two. We're going to copy this one through all three, but I'm going to add one in the middle. This one is L.16.17, compare and contrast mitosis and meiosis and relate to the process of sexual and asexual reproduction and their consequences for genetic variation. And so the goal here is to understand why we go through mitosis, why we go through meiosis, what's the difference, and what do they produce. So here is a typical stages of human development. You know, you start out as an infant, then a toddler, then an adolescent, then basically a young person in your 20s and 30s. Then you start getting older, and then older, and finally old. And what's the difference here, cellular wise? I mean, what's happening as this person develops and grows? What causes this to develop, and then what causes the aging process? And it all has to do with cells. Everything that you see happening here is based on cells. And so it's the growth of the cells, the development of the cells, and what the cells do for the body that we're going to study here. So the first thing I want to ask you is this represents time, right? I mean, this is this same person, but as an infant, a toddler, an adolescent, a grown up, and a middle-aged person, old man, and a very old man. What happened to their bodies, and what does that have to do with cells? So the first thing I want you to know is that from here to here, what happened in terms of cells? And I want you to think about this question. Did the cells that make up this baby get larger to create this thing, and then get larger to create this, and then get larger to create this, and this, and this? Or did this thing just add more cells and add more cell and add more cells? So let's talk about that real fast. Cell division versus cell growth. Cells do grow. They get larger in size, and they also divide. So the difference is that a small cell becomes a large cell through time. As it grows and matures, it absorbs fluids and other things, and it gets larger, because when they're first created, they're somewhat smaller than normal. They have to get bigger, but that same cell can also divide and then make these smaller cells. So what happened from the toddler, or from the infant to the toddler? Did all of its cells get bigger, or did its same cells just grow and divide and then get bigger? Sorry about that. The answer, of course, is that it did a little bit of both, but mostly what happened is that the cells that made up this toddler divided to make more cells. And so this organism here, this toddler has more cells than this infant. This adolescent has more cells than this toddler, and this adult has more cells than this adolescent. And as you are developing, one of the things that you're doing is you're adding cells. So cells that you already have, because remember, one of the parts of the cell cycle is that cells only come from pre-existing cells. At some point, you can trace this all the way back to a single cell. Okay, a fertilized egg, half mom, half dad. And then that cell grew and divided, grew and divided, grew and divided, and then started to differentiate. If you paid attention to another set of videos where we talked about that, this thing started to differentiate, and then the cells started to become skin cells, bone cells, blood cells, hair cells, eyeball cells, all those different kinds of cells. And eventually it became a structure that could survive on its own, and the baby was born, and then the growth continued. And throughout, from the moment that egg was fertilized by that sperm, it started to grow. Well, actually, at that moment, it wasn't growing, but it started to divide, and then it divided and divided and divided. Then it started to get nutrition, and those grew, and then grow and divide, grow and divide, grow and divide, until it gets to the point to where you've reached full maturity. This is what we call an adult. Now, this adult is probably not going to gain much more in size, probably a little bit fat around the middle, but in terms of development, they've reached full physical maturity here, and that's all done through cells, and the fact that the cells grow and divide and add more cells to make the body larger, and those cells also differentiate to develop different tissues and different structures inside of the organism, and it reaches full maturity. And the aging process starts from the moment the egg is fertilized. But from then on, the body is not interested in growing anymore. It's simply interested in maintaining. And over time, its ability to maintain, which is done through production of cells, over the time its ability to maintain is diminished because the body's getting old, and after a while, the ability to repair and diminish and the ability to repair and maintain its systems is diminished because of um, its the age, and finally the body dies. And this is a natural process. It happens to everybody. But keep in mind, all of it: growth, organization, repair, development, all of that is done simply by adding cells. Cells grow and divide and create new cells, and that's where we get all these processes. And this is important to know. So we're going to study these processes of cell growth. We're also going to study what happens when cell growth goes wrong. And then we're finally going to study how we get that fertilized egg and sperm to start with, so that we can, at this point, reproduce to create a whole new generation. Okay? So this particular video, part one, is just going to be about how cells grow and divide, and it's a process. Okay, cell division is a process by which new cells form from existing cells. One cell divides and becomes two, two becomes four, four becomes eight, eight becomes 16. And in between each one of those divisions, there is a life cycle. It's growing and dividing and growing and dividing. One cell grows and divides and becomes two, two cells grows and divide become four, four becomes eight, and they'll grow as much as needed. And when you're young, when you're at this state, you're never growing any more than you are that day. You're growing like crazy. That's why babies have to eat so much and sleep all the time, because their bodies are just focused on growing. And then at some point you reach kind of puberty stage, and your body says, "Oh, time to do it again." I mean, you kick in and start growing, and that's why you eat like a horse, because you're needing so much nutrition and just simple mass to make your body larger. And then your metabolism slows down, and it's like, okay, we've reached our full physical maturity, so now we don't need to eat so much, and we don't need to develop so much. And so, your processes, you're in your pretty much your prime right there. And then it's downhill from there. And sadly, I'm somewhere between this guy and this guy, and my body has my metabolism used to be something I could eat like crazy, but now I still eat like crazy, but I'm no longer using that much energy. So guess what happens? My body makes fat. But anyway, the point is that it's all based on these cells. Now, there's two kinds of cells. There's prokaryotes and eukaryotes, and they divide differently. Prokaryotic division is very simple. We're going to show that to you right here. This is called binary fission. But then eukaryotic division is a little bit more difficult, because it's got a lot more stuff inside. Remember, it's a more complex cell. And so it's got a lot more stuff inside, especially the nucleus. And the nucleus dividing is one of the most important things that happens during the division of a eukaryotic cell. And then finally, there's another type of division of eukaryotic cells called meiosis. And this is really important, because it's got to divide up all the DNA and everything and to create sex cells, which is really complex and requires a lot of well, it's crazy that it actually happens and works. Okay, but let's talk first about binary fission. It's a very straightforward process. A prokaryote is an unbelievably simple cell, so it's not that hard. It's no big of a deal. You got a cell, you got DNA inside of it. The DNA makes a copy of itself. One copy goes this way, one copy goes this way. The cell splits down the middle, and bang, now you got two cells. It's important to note that these two, what we call daughter cells, are genetically identical to the parent cell. These two daughter cells are just this big cell split in half, but this happens during both binary fission and mitosis, same kind of a thing. In fact, mitosis is very much like this. The only difference is that this process here of copying the DNA takes just a little bit longer. So this is really how mitosis works. Just something, some extra added special stuff in here, because the DNA gets so complex. And also inside of here, there's very few things that need to be copied. So putting half the stuff in here and half the stuff in here isn't that big of a deal. Whereas with eukaryotic cells, there's a lot more stuff in here, and you have to have half of it goes into the cell and half of it goes into the cell. It takes longer and just requires more work. But this is pretty straightforward. Again, here's the DNA. It gets copied. Right here, it's already copied. Okay, one goes this way, one goes this way, the cell splits down the middle, and wada bing, you got two new cells. This is binary fission, really simple stuff. But when you're talking about eukaryotic cell division, we actually have a life cycle of the cell. It's a lot more complex. I mean, it is the same process, and you can think of it as just an advanced form of this, because that's all it really is. But from before we copy the DNA, or from the time the DNA gets copied all the way through division, There's a lot more interesting things that go on, and so we want to talk about that. We divide it up into these phases. It has this life cycle, and for no real reason, we're going to start with a G1 phase. And the G1 phase, because with all cycles, where do you really start? But in this case, we're going to start with a G1 phase. That's the first time that the cell has become itself. It is right at the end of cell division. And the cell, actually, G0 is right at the end of cell division. We're going to skip G0 for a minute and go to G1, where it starts to grow. That's what the G stands for in growth, or in G1. It's growing. The cell grows. Remember, I told you right up here. The cells grow, right? And so the cell's going to grow. And how much time? Nah, it's hard to say. But the cell's going to spend some time in growth, and then there's some point there's going to be a signal where it stops growing, and it starts making a copy of all of its DNA. This is called the S phase. Inside of here should say the word replication, because this is where DNA gets replicated. Now we haven't talked about that yet, and that is a huge process. Notice how much time the cell spends on it in its life cycle. Okay, and by the way, this isn't the cell. This is a kind of a flow chart or a timeline in a circle format. Okay. The S phase is a significant amount of time in the life of a cell because it takes so much time to copy the DNA, and we haven't gotten there yet, so put that in the back of your head. We're going to be talking about copying DNA very soon in a process called replication. But that happens here during the S phase. Basically, what happens is at the end of the G1 phase, I have one copy of DNA. At the end of the S phase, I have two copies of DNA because I've made a copy of everything. So now I go into the G2 phase, and you can say one copy and two, although that's not what these mean, but that may help you to remember that there's one copy of DNA here, and now there's two copies here. But in the G2 phase, that just really means the second growth phase. The cell does more growth. Okay. Now also during the G1 and G2 and G0 phases, the cell is doing its job, whatever its job is. If it's a skin cell, it does its skin cell work. If it's a blood cell, it does its blood cell work, although blood cells don't quite work like this. But either way, in the G0 phase, there's no growth, it's just doing its job. Okay, so during the G phases, the cell is doing what it does. The brain cells, the bone cells, whatever they do, right? All the work stops during the S phase so it can copy the DNA. Okay, but then it goes back to working and grows more in preparation to do this thing, the M phase, which is called mitosis, and then finally to split in half and become two new cells. The goal here is that depending on the time of the life, as needed, these one cell can split and become two. The goal here is to create two new cells, both of them identical copies of the original cell, because that's how we get more cells. Okay, and that's how we grow, that's how we develop, that's how we repair. If you get cut on your skin, the skin in between the cut, immediately those cells start doing this. They start creating more and more cells so that they can fill in the gap. That's how they do it. They fill in the gap by growing more cells. You get a scab, the scab stops the bleeding, and then underneath the scab, the cells start growing, and then they can connect back together. And next thing you know, you can't even tell you were cut there. But this is that process. It doesn't do it by some kind of a weird glue or some kind of an amazing process. It's just very simple. Hey, there's an empty hole there. Now we're just going to grow cells and fill it up. Okay. This process is called mitosis, and like I said, it happens in your body literally thousands of times a second. It's unbelievable how much this happens in your body. Okay. We're going to spend a lot of time talking about the M phase and a little bit about cytokinesis, but during the M phase, this is a very important thing. The nucleus divides. Now remember, the nucleus is where the DNA is, and at the end of the S phase, now I've got two copies of DNA, and the goal here is I've got to get a perfect copy into one cell and a perfect copy into the other cell, and that takes a lot of work. It takes a choreographed set of steps to ensure that each cell gets an exact number. If you get more of one in one than the other, then they're going to die, and that creates a real issue. So there's a lot of time spent in the M phase to ensure that the correct amount of DNA ends up in each cell. An exact copy, the exact amount, exactly half goes this way, exactly half goes this way. And then cytokinesis, of course, is the end of this process in which the cell splits in half. That's what cytokinesis means. It means split cell. And then process starts all over, brand new bouncing baby cell. It grows, replicates, grows some more, and does it again. Okay, how long this takes can depend. Sometimes when you have like if you cut your skin, then your cells need to be growing and dividing very rapidly. So sometimes they go right from cytokinesis right back to G1, and they go it again and again and again. But at some point there's a signal that says, okay, we're good. And then the cell goes into the G0 phase, in which it stops growing and it just does its job. And it'll stay there until there's a signal that says, oh, hey, we got to grow some more. So it goes back into its G1 phase. Now, I want you to remember that particular step about the start and the stop signal, because that's going to come into play in video number 2. All right, this is a continuous process, but like I said, the amount of time just depends. A cell can be in a G0 phase for a very long time, years, or in a very brief time, literally seconds, before it goes back into the G1 phase, just depending on the need. And when you're young, when you're an infant, or still an embryo inside of your mom, then this process is going to be happening faster, because that's what you're doing, is you're just growing and dividing cells. I mean, I'm going to go back up here real fast. I mean, from the fertilized egg all the way through, that's what's happening, is cells are growing, dividing. Now it slows down a little bit, you know, and then it speeds up a little bit at puberty, but it slows down a little bit. But the goal here is to get here to this full physical maturity. Okay, and so the way that you do that is you just keep adding cells and keep replacing dead cells, because we have cells die all the time. And this is how we replace dead cells. This is how we add cells. This is how we repair our body. This is how we replace dead cells. I mean, every time you scratch your skin, dead cells are falling off. How do you get new cells? Through this process. This is how we grow. This is how we develop. This is how we repair. It's through this exact process. Okay. There are 3 main phases. Everything that's not cytokinesis and mitosis. So the G0, G1, S, and G2, these are all something called interphase. And it just means the phase in between. All right. And it's the phase in between division. So from G0 to G1, S, and G2, that's interphase. That's where the bulk of the cell life is spent. But after a while, when it's time to split up, the mitosis begins. That's the mitosis phase. And then there's a cytokinesis phase. The cytokinesis phase is very short. In fact, it starts as telophase is ending. You'll see that in just a minute. But these 2 phases tend to happen together because, again, efficiency and speed is what the process here is. So you don't wait until telophase is over to start cytokinesis. You actually do them together, kind of. And you'll see that in a minute. Okay. And this is everything I just said. So let's take a quick look at the parts of the 3 parts of interphase, not including G0, because remember, in the G0 phase it's just sitting still and doing its job. But the G1 phase is cell growth. The cell grows, it repairs any damaged organelles, it increases in size. It does carry out its routine functions, it takes nutrients and uses them for energy, but it is growing. That's what's different than the G0 phase. In the G0 phase, it's not growing, but in the G1 phase, G0 looks just like this, except it's not growing. G1 is the same as G0, except that it is growing. Now, at some point, the growth stops, because growing takes energy, and we now need to take all of our energy and do the S phase, which is to copy the DNA. And we're going to take a real close look at what happens during the S phase in a couple of videos a little bit later on. But suffice it to say that I have one set of DNA at the beginning of the S phase, and I copy all of that to get a full set of 2 full sets of DNA at the end of S phase, and it has to be done exactly right. If there is a mistake, there are 3 billion pairs of DNA that have to be copied during the S phase, so it has to happen at an unbelievable speed, literally jet speed. Okay, but think about this. If I make one mistake, if I screw up one copy, I now have a mutation. Here's a good analogy. Suppose I gave you a fairly thick book. Think of a textbook, and I said, here's blank sheets of paper. You have to copy it exactly. Okay, can you imagine how difficult it's going to be? And don't worry about getting tired. But can you imagine if you didn't get tired, but if you had the responsibility of taking every letter and every picture and everything, let's just say letters, every letter from the textbook, and make a new copy of it? What are the odds you're going to make a mistake? Pretty good odds, right? You can't do that if you're a cell. Okay, and the other thing is this. The next thing I'm going to tell you is, oh yeah, you got 5 minutes to do it. Holy crap, how do you do that? The cells are very good at doing this, and they do this at a high rate of speed. In fact, they do it so fast that they have time to go back and check. After they get through making the copy, they go back and check, and they go, oh, look, Doofus, you made a mistake here. They erase it and fix it. And that all happens during the S phase, and it happens at an unbelievable rate. Okay? And yet, they still make mistakes. Mutations get through. In fact, it's been estimated that every time a cell divides, a mutation happens, and that mutation is what leads to one of the things that contributes to things such as cancer or aging. In fact, it's been argued that aging is nothing more than a buildup of these mutations over time. Then when the S phase is over, then the G2 phase starts again. Again, just like the G1 phase, except remember that in the G2 phase I've got two copies of the DNA and I'm growing more. I'm adding extra organelles. I'm trying to double up my organelles and everything, trying to add in some cytoplasm because I'm about to split this whole thing in half, and I need enough organelles for now for two cells and enough cytoplasm for two cells, so I got to do all that. So the cell gets a little bit bigger than normal, kind of plumps up during G2 phase because we're about to split it in half. Okay, but remember, all the G phases are the same. The cell is doing its work. The only difference is in G1 and G2 it's actually growing, whereas in G0 it's not. Alright, moving on. Mitosis is a process of splitting the nucleus, and that's what mitosis literally means. It has four phases: prophase, metaphase, anaphase, and telophase. I think I would like to show you this graphically, but I'd like for you to maybe pause this and read it. But it all has to do with taking all that DNA and moving it in such a way that it is going to move correctly, get the exact number into each new nucleus, because remember we're splitting the nucleus, so we're making two new nuclei, and each one of these nuclei has to have the same amount of DNA, the exact amount of DNA, right? It has to split it up. So this process, prophase, metaphase, anaphase, and telophase, is how that's done. It's a really interesting process, and I'm going to tell you, but the first thing I'd like to tell you is this: we're coming up on 20 minutes, and I figure I've got at least 10 more, so you may want to pause and take a break. Alright, if you just paused and take a break, welcome back. If not, well, whatever, dude. But, so prophase is the first process. DNA normally is not found as chromosomes. Remember that chromosomes and DNA are the same thing, but chromosomes are DNA all packed up. Here's my analogy. I moved recently, and I had to, pack up all of my stuff and move it. Now, I like to eat, so I need dishes to eat on, okay? But the problem is I can't just take my dishes and throw them in the back of the truck because they'll break. So I have to pack my dishes up nice and careful and put them in a truck so that I can move them, and then as soon as I get to my new home, I unpack them and use them. Well, the thing is while they're packed up, they can't be used. So did I drag my feet while I had my dishes all packed up? Absolutely not. I packed them up neatly and carefully moved them as quickly as I could, and then unpack them so that I can use them again. DNA is the same way. Normally it's in the form of something called chromatin. Chromatin is DNA, I kind of call it all fluffed out and unpacked. It would look kind of like cotton inside of the nucleus. But I can't move that chromatin because it's big, it's easy to break, and it just doesn't work well when I try to move it that way. So it packs up. I'm going to show you that on another video. The chromatin, which is DNA in its unpacked form, packs up into a chromosome. Now, a chromosome is a solid, tight structure. Now, the DNA can't be used, so we don't like to have our DNA in chromosomes any longer than we have to. But when I put these DNA into the chromosomes, well, now they're packed up, and now they're ready to be shipped. Now, if you remember, the prophase happens here. That's well after the S phase. So now I've got two copies of the DNA at the beginning of prophase, and they're paired up. So if I had you remember chromosomes are structures, and in humans we have 23. So let's just pick one. Let's say chromosome 17. Back in the G1 phase, I had only one chromosome 17. It's all unpacked, but I only had one of them. But it got copied in the S phase, and now here I have two chromosome 17s, and they're called sister chromatids. Okay, and these sister chromatids kind of attach to each other, right? And they're attached to the regions called centromeres. This helps to make sure that they line up and they get split apart evenly, because I want one chromosome 17 to go into one nucleus and one chromosome 17 to go into the other nucleus, and this is the way that happens. Also, during the prophase, the nuclear membrane breaks down. Remember, all this is happening inside the nucleus, but now I need some room to work. And so the nuclear membrane breaks down and kind of moves out of the way so that I can have the whole cell to do this job. These centrioles, which are kind of like cranes, and spindle fibers, which are kind of like the cables on a crane, they start to form and they start to attach themselves to these central regions of these chromosomes, or these sister chromatids. Okay, I'm going to show you all this again in well, let's just do it here. In prophase, it's hard to see, but the nucleus is starting to break apart. You can start to see the chromosomes where they're packing up, and here I have these centromeres and spindle fibers, and these spindle fibers starts to stretch out and to attach. Okay, now that's what happens in prophase. If you want to identify prophase, you'll notice that these spindle fibers are starting to form, and this in the DNA, in the chromosomes first form. Okay. Now then, in metaphase, something really interesting happens. These centromeres, these centrioles and these spindle fibers are moving the chromosomes around to where they line up evenly at what we call the equator of the cell. And if you think about it, if this is a cell and this is a north pole and a south pole, then right down the middle is the equator. So these spindle fibers are moving all these chromosomes, and remember there's two pair there, lining them up right there. Okay. It's actually pretty brilliant how they do this. This is going to ensure that half the chromosomes end up over here and half the chromosome, a full set, one single set. Remember, I got two sets here in the middle. One set's going to end up here and one set's going to end up here. Okay, that's what happens during metaphase. Well, in between, there's kind of prophase, prometaphase, and metaphase. Normally, we don't talk about prometaphase. So, at the end of metaphase, the chromosomes are lined up here. Okay, and I got two full sets and they're lined up at the end of metaphase. Now then in anaphase, these spindle fibers contract and they start to pull on the sister chromatids. Okay, and remember the sister chromatids. These are the pairs, right? These are the this was the two copies attached to each other. Okay, and they line up right here at the equator in metaphase. And now when these spindle fibers contract, they start pulling. And so now this is chromosome 17 right here. This is just, I'm making that up, but this is chromosome 17 right here. There's one copy on this side and one copy on this side. And when these things contract, right smack in the middle at that centromere, they pop in half, and one of the chromosome 17s go this way, and one of the chromosome 17s go this way. And that happens in humans for all 23 chromosomes, light up right there in the middle. And so anaphase is a really crucial phase because this is where the DNA splits in half, and half of it goes this way and half of it goes this way. And this is how we're going to ensure that a full set of chromosomes end up over here and a full set end up over here. Now remember, the chromosomes are all packed up right now. We can't use them, and the cell's like, look, I got things to do, we got to get this done. So it's happening fast, but it has to happen fast and it has to happen accurately. And right here during anaphase is where some bad things can happen. If a spindle fiber breaks, and instead of the centromere splitting in half, if two copies end up over here, then you got a problem. The cell's going to die. So it can't screw this up, but it has to happen fast. Does that make sense? There's lots of situations where it can't screw it up and it has to happen fast, and believe it or not, it happens correctly, 99.9% of the time, or probably better than that. It's an amazing process. That's during anaphase. Anaphase, these things are starting to head towards we call these the poles. These things are starting to head towards the poles. I've got one full set of DNA here, one full set of DNA here. Okay, and notice that right here in the middle, everything's. Notice also all the other stuff like the cytoplasm and all of the other parts of the cell, the organelles, they're headed this way too. Okay, and then during telophase, these things get to the poles, the nucleus forms around them, they start to unpack back into chromatin, and that's telophase. And what it's not showing you here is that there's starting to be a line drawn down the middle, and then we're going to start cytokinesis. Okay, so cytokinesis is this process to where now I'm towards the end of telophase. The chromosomes have reached each side. I got a full set here and a full set here. The nucleus is starting to form around them, and the cell starts to pinch in half. This is literally, I promise you, I'm not lying when I tell you this is called cleavage. Okay, and as the chromosomes unpack back into chromatin, the nuclear envelope or the nucleus, the nuclear membrane forms around them. The cell splits in half, and now I literally have two new cells identical to the original cell, and this is the goal. These go back into the G0 phase, and they both start the process all over again. If this was a little confusing to you, I would encourage you to go back and look at it again. I'm going to take it through you one more time here briefly. Here I have one original parent cell. At the end of G2 phase, it has two copies, or I'm sorry, I shouldn't say it that way. It has two full sets of DNA, enough to make two cells. Okay, the DNA is thickening up, so this is really at the beginning of prophase. By metaphase. This is pro-metaphase. They're starting to line up. By metaphase, they've lined up. By anaphase, they've split in half and headed in each direction. And then by telophase, they've reached the poles. The nucleus has started to form around them again, okay? And they're going to split down the middle. The DNA is starting, the chromosomes are unpacking back into chromatin, and then right there in the middle, pink, I've got two new bouncing baby cells. They go back into the G0 um, part of the cell cycle, and they do their job. So I've taken one cell and I've created two. And this is how everything is done in your body in terms of growth and development and repair. And it's happening in your body like a zillion, quadrillion times a day. Okay? And it's a good thing. And it has to be done right. Remember here during the anaphase, it has to be done right. And and it has to happen constantly. And in fact, aging occurs because this starts to happen less and less, and it starts to kind of get some mistakes in it with the buildup of those mutations. And we don't do quite what we used to be able to do when we're younger. Trust me, it hurts. Try not to get old if you can avoid it. But that is the end of video number one on mitosis. I need you to understand the goal here. Take one cell, make an exact copy of that cell into two new cells. Okay? And these will go through the cell cycle again, and they'll become two more later on, and then though that they'll become two more, and they'll become two more, and this is how we get all of our body. Your body went from one cell to quadrillions of cells through this exact process. Okay, watch this again if it's confusing, but remember it's supposed to be a review. Okay, that's it. Have a good day. Get to work.